So here we'll discuss how chromosomes are maintained, including mismatch repairs, centromeres, and telomeres. So sometimes errors happen during replication that aren't detected and corrected by the proofreading function of polymerase. And so mismatch repair is the process of reversing those errors. And having correctly functioning mismatch repair is really important for maintaining genomic stability. So first, mismatch errors produce these heteroduplex structures or bubbles in duplex DNA, and that's because with the improper base pairing, the nucleotides don't bind together. And so these heteroduplex structures are recognized by a protein complex of specific factors in the mismatch repair pathway. And so these are their mismatch recognition proteins. And so this complex of bubble recognition factors then recruits additional factors that identify the most recently synthesized strand. And how it does this isn't quite clear. But once the most recently synthesized strand is identified, the mismatch containing strand is then removed by an exonuclease from the point where it was recognized as recently replicated all the way to the mismatch. And the amount removed is variable, and it's frequently pretty long, typically between 300 to 500 nucleotides. And so now there's this intact single-stranded DNA, and that template strand is then used to direct gap repair. And it's a process that's similar to the resolution of the Okazaki fragments. So the loss of function of one or more of these mismatch repair proteins leads to an impaired ability to repair DNA. And this can lead to the accumulation of mutations across the genome, which affects the function of many genes, including genes that control cell growth. And so Lynch syndrome is one of those conditions. And affected individuals are at increased risk of developing one or more of a range of different types of cancer. And this was originally termed hereditary non-polyposis colorectal cancer because it was commonly associated with colorectal cancer. But later it was recognized that it was actually associated with a broad range of tumor types and was renamed after the physician who first described it. So in addition to DNA repair, a couple of other processes contribute to the proper maintenance of chromosomes within cells. And they are the segregation of chromosomes during cell division, and that relies on a structure called a centromere, and the protection of the chromosomal ends, which relies on telomeres. And so this image is of a chromosome in metaphase that has been duplicated. So there are two sister chromatids with the centromere joining them and purple telomeres on the end of each chromatid. And so if we first look at centromeres, they're important for guiding the chromatids to the daughter cells during cell division. And the placement of the centromeres relative to the length of the chromosome is different for each chromosome. Centromeres can be in the center of the chromosome, like the name suggests, or closer to one end. And so centromeres consist of some defined sequence elements, but they're mostly less well-defined repetitive sequence. And in multicellular eukaryotes, centromeres are typically several 100,000 base pairs and include an alpha satellite repetitive sequence. And then there are the telomeres that protect the ends of the chromosome from erosion of gene sequences, and they're regions of repetitive nucleotide sequence at the ends of the chromosome. So first, let's look more closely at centromere function and how centromeres control the segregation of chromatids during the mitosis phase of the cell cycle. And so remember that just before cell division, during the S phase or synthesis phase, DNA replication generates 2 copies of each chromosome. So in a human somatic cell prior to DNA replication, so in this stage, we would have 46 chromosomes and 46 chromatids. Then after DNA replication, we would have 46 chromosomes still, but 92 chromatids. And so during mitosis, these chromosomes line up and the chromatids separate, with the end result being 2 identical daughter cells. And the centromere is really important here for the organized segregation of those chromatids that happens during mitosis. And so that's because during mitosis, the spindle fibers attach to the centromere, and the spindle fibers are part of a protein machine that causes this series of steps to happen. So with the duplicated chromosomes lining up in the middle of the cell, then splitting the chromosomes into their individual chromatid copies, and finally dragging one of each copy to each of the 2 poles of the dividing cell. So each daughter cell ends up with one and only one of each of the copies. So the centromere is really the basis for segregation, and the rest of the chromosome just follows that centromere. And so if segregation isn't done properly, it could cause the wrong number of chromosomes in each daughter cell. And cells with the wrong number of chromosomes are called aneuploid, and aneuploidy is commonly seen in cancers. Now let's look at telomeres and see how they contribute to the maintenance of chromosomes. So the ends of a linear chromosome cause a couple of major problems that have to be overcome. So first, there's this end replication problem. So initiation of replication requires a primer. And once initiated, the primer is removed, and that region is replicated by a polymerase that initiated replication further downstream. But unfortunately, on the end of the chromosome, there is no further downstream site of replication. And so as a consequence, a short amount of sequence at the end of the chromosome is left unreplicated after every round of replication. And so if this proceeded indefinitely, then genes near the end of the chromosome would just get deleted. The second problem is that the DNA ends are sensitive to degradation. And so they're seen by the cell as a form of DNA damage. So as a result, the chromosome ends could trigger a repair type of reaction where the ends of 2 chromosomes are fused together. And so fusion of chromosomes would mean that they couldn't separate properly during mitosis. Or the chromosome end could trigger cell death through checkpoint pathways, and really either one of these pathways would be unfavorable for an otherwise healthy cell. And so these problems are solved by telomeres. And a telomere is a region of repetitive nucleotide sequence at the end of the chromosome, and early in development in actively dividing cells, several telomeric repeats are added every round of replication by a specific enzyme named telomerase. And so this opposes the loss in telomere sequence due to that end replication problem. So in other words, the eukaryotic cells balance the loss of telomere sequence due to the inability to complete replication to the end, with the addition of new telomere repeats through telomerase activity. And so telomerase really resembles a standard DNA polymerase in that it adds new DNA to the 3' end of a growing chain. But unlike standard DNA polymerases, telomerase doesn't use a DNA template to direct synthesis. Instead, it uses an intrinsic RNA component of the enzyme to act as the template strand. So when telomeres are viewed by electron microscopy, structures known as T-loops can be seen. And this loop is where a single-stranded tail at the end of the telomere is looping back and inserts itself into the region of the double helix. So here's our single-stranded tail, which is wrapping around and inserting itself into the double helix. And so this T-loop was discovered in 1999 by Dr. Jack Griffith at UNC. So his lab found that formation of the loop was mediated by the telomere repeat binding factor 2, or TRF2. And so this loop essentially hides and protects the end of the chromosome so that the end of the chromosome is resistant to degradation or any attempts at repair. And telomeres are important in both aging and cancer, and there's a clear link between the potential of cells to regularly divide, or their proliferative capacity, and the length of their telomeres. So cells with short telomeres have a limited proliferative capacity, and cells with long telomeres have much greater proliferative capacity. And so this leads to the idea that telomeres are like a clock that helps to define proliferative capacity. And with every cell division, the telomeres shorten, and after a certain number of divisions, called the Hayflick limit, which varies depending on the cell type, the telomeres have shortened to a point where a signal is sent that stops the cells from dividing further, and this is called senescence. So cellular senescence is a significant component of aging. So you can kind of think of telomere length as acting as kind of a measuring stick to count the cell divisions and regulate the lifetime of the cell. And so telomerase, that enzyme that adds the repeating segment onto the ends of the chromosome, so early in development, cells stop expressing telomerase. So the cells continue to divide, but because they're no longer expressing telomerase, their telomeres shorten with each division. And cancer-causing mutations can force this type of differentiated cell to start dividing again, and this process is known as transformation. But again, the telomeres continue to shorten even more with each division. The transformed cells eventually reach a crisis point, and that's where the telomere becomes too short to allow for cell survival. And although most transformed cells are killed at this point, some survive and go on to cause cancer because they've learned to re-express telomerase, and their telomeres no longer continue to shorten. And this involvement of telomeres in aging and cancers has been supported by lots of research, but one example is that when mice are bred to entirely lack telomerase, they show signs of premature aging and have a tendency to develop tumors.